Welcome to the first day of A&P. So we are going to start with body chemistry. Some of this stuff is kind of basic review. Some of it might be things that you maybe never saw or never really understood before. So that's going to be our goal today, to lay a foundation for what we're going to talk about as we go along. But one of my main goals with A&P here is to give you information that's actually relevant to your jobs. If I learn something and I don't recognize how it's significant, I typically won't retain it, or I won't listen and I won't care. So I do want to talk about with A&P as we go along how this stuff actually matters to what we're doing in practice with our actions as, treating patients for different diseases, or maybe helping them to be explained to the clients and things along those lines. So I do think most of this stuff is directly relevant to what we do, and I'll try to point that out whenever possible. But if there are times where you don't understand why we're talking about something, please do let me know, because I want to make sure that it feels like it's at least relevant to your life. In middle school or in elementary school, we all learn that there are two types of cells, our prokaryotic and eukaryotic cells. Prokaryotic cells are the more primitive cells. They evolved first, and they're very simple and very small. So this is going to be cells like our bacterial cells. We've talked about in lab at some point, right? A red blood cell is a big cell. It's a eukaryotic cell. It's an animal cell. And bacteria, by comparison, is very, very small and does not contain the organelles or the nucleus. Eukaryotic cells are going to be all animal cells, okay, and all plant cells. So these are more complex. They contain organelles, so that's things like our mitochondria, or plants, our chloroplasts and ribosomes and all that stuff. And we're not going to go over that again because I don't really think that matters, at least not in detail. One of the things I do think is super interesting about the development or the evolution of these cells is that they think, and we have good proof now, that the way this system developed, the way that eukaryotic cells evolved, was that there were two prokaryotic cells. Right? Maybe one was slightly bigger than the other, and the little guy was like, hey, can I go ahead and hide inside you? And the bigger cell said, well, what are you going to do if I let you hide inside me from, scary predator out here? Okay, another prokaryotic cell. And the little guy said, well, what if I give you energy, or I give you glucose, or I give you some substance that I can produce? And the big guy said, okay, I guess we can do that. And that's how we got eukaryotic cells. So you might have heard that mitochondria have their own DNA. And it's true, they have at least their own genetic material, and that's because originally they were a separate organism. So kind of an interesting relationship there for sure. But that's how we think organelles developed in these eukaryotic cells. So all of our cells in our bodies are eukaryotic cells, and that's what we're going to focus on here. So your eukaryotic cell structure, we're going to have a nucleus. Some cells do lose their nucleus at some point during development, like those red blood cells that we've looked at. We've got endoplasmic reticulum. These are usually in charge of packaging proteins and so forth. And then mitochondria are what we're going to focus on for our purposes. So mitochondria are the organelle in the cell that's responsible for cellular respiration, which is producing energy, taking in energy from the outside, turning it into some form that we can actually use, and essentially keeping us alive. You can think about them as, the engine or transmission in your car. You need them to be able to go. The cell needs them to work to be able to live. And this on the left here is something that you've seen before, probably memorized before at some point, which is the Krebs cycle. We're not going to memorize this again because this is not super relevant to our lives here, but what we really do want to understand is that for cells to survive, okay, they need oxygen and glucose. So when we start to look at things like the circulatory system, what's the job of the heart? The job of the heart is to move blood around the body. Why do we care? It's because the blood is carrying oxygen, and if the cells don't get oxygen, mitochondria can't do this cellular respiration to make ATP. And ATP is adenosine triphosphate. Okay, and it is the currency. It is the energy that is needed to do things like maintain homeostasis, which we're going to look at shortly here. So ATP is required to be able to do these things. Now, sometimes we do have other ways, or we can temporarily make ATP without oxygen and glucose. So many cells have the ability to operate, at least briefly in our bodies, anaerobically without oxygen. But when they do that, we get nasty waste products as a result. Sometimes we've talked about in other classes, if cells in our bodies have to switch over to anaerobic metabolism, they start to make lactic acid, right? That's the substance that causes your muscles to burn if you run up and down the stairs. Same with glucose. Cells can operate for a period of time without glucose, but they'll suffer for it. And we'll see, for instance, our brain cells in particular, all of our nervous tissues have a very high requirement for both oxygen and glucose. But if we don't get enough glucose, our brain cells will shut down, we'll start to have seizures, and we'll die. So we need oxygen, we need glucose going to our cells at all times, and all of the other organs in our body are responsible for maintaining this demand of the mitochondria. So that's how I like to think about it. Another feature of cells that we care about is the fact that they have a membrane on the outside. So all of our prokaryotic cells, all of the cells in our body do have some kind of defined membrane for most of their life cycle anyway. And this is selectively permeable. That means that it's going to let certain things in and certain things out. Some things are going to be able to get in really easily. We're going to see that water moves pretty readily back and forth across the membrane, but other things like, let's say glucose, are going to require help. Okay? They're not going to be able to move without some sort of assistance in and out of the cellular membrane, or in and out of the cytoplasm of the cell. So we can see situations where glucose is a really good example. Let's say we have a diabetic patient and they don't have enough insulin. That is diabetes, right? Diabetes mellitus, our pancreas doesn't make enough insulin. Well, what does insulin do? It actually opens one of these little channels here. It opens a spot in the plasma membrane so that the glucose can enter the cell. In the absence of insulin, okay, glucose can't do that. And so there are definitely situations with electrolytes or with other substances and proteins and things like glucose in the body that our cell membrane plays a role. And we have to figure out how to get stuff in the cell in order to balance out the system so that the body can operate properly. The cytoplasm is, of course, all that fluid inside the cell that contains the nucleus and the other organelles. And our goal is to make sure that the appropriate amounts of proteins and electrolytes and glucose and oxygen are available in that cytoplasm so that the cell can do its job. Remember, we said its job is make ATP so that all the other chemical reactions in our body that we need to stay alive can happen. So that's our goal. If you're ever taking care of a patient in the hospital and you're like, what am I actually doing here? This is what you're doing. It's kind of crazy to think about it that way, but it helps me to remember this stuff. Glucose, protein, electrolytes, and oxygen. In terms of that cell membrane and moving across it, there are a few specific ways that we need to know that things can get into the cell. And water's in its own little special category. We focus a lot on the behavior of water, but water moves by what we call osmosis. We only use this term osmosis to refer to water. And osmosis is actually just the simple diffusion of water, and this is going to be with the concentration gradient. So what is the concentration gradient? Well, moving with the concentration gradient means that water is going to move from the area of low concentration to high concentration of the solute. A lot of the time the solute, when we use this term solute, we're talking about specifically sodium or salt. Okay, this really drives the movement of water more than any other electrolyte, but theoretically it can be anything. Typically we mean salt here. So the concentration of salt on this side, you can see in this image, is a lot higher. Let's say each of these is a salt molecule. Well, what does water want to do? It wants the concentration on both sides to be balanced. So if I took a drink from this side and I took a drink from this side, Okay, they would taste different, right? This would have like a weak salty taste. This would have a very strong salty taste. And water moving with the concentration gradient means it's going to want to dilute out. It's going to want both these sides to taste the same. Okay, or basically we're saying they should be the same saltiness. And sometimes we use the word instead of saltiness. We'll see later in the presentation here, tonicity. You've probably heard that one before in reference to fluids. But that's all it means here is the same saltiness. So water in this case, if we have our semi-permeable membrane, is going to move with the concentration gradient. So it's going to move towards the saltier side, so that now if we take a drink from this side and we take a drink from this side, they're going to have the same level of salt there. So that's the concentration gradient. And what does that mean for us? It means that we can influence the movement of water by changing the solute concentration. We'll talk about that a little bit more as we go along. It also means that water, for the most part, does not require energy to move through the body. Really easily it can move in and out of all these fluid compartments. So when we do something like give IV fluids, they don't stay for very long in the intravenous space. They move really readily elsewhere because osmosis is a really simple basic process. Water really easily moves across these cell membranes and tissue separations, and it doesn't require any energy to do so. We're going to look at the three other forms of transport that we care about here. The first two are passive methods, so this means they don't require ATP, and the last is active transport. There's a few different kinds, but all active transports do require ATP. So that's how we differentiate between passive and active. Now diffusion, we're going to see, works well for small molecules, especially gases. Water, we said already, works by diffusion. We just call it osmosis. It's in its own little special category. Facilitated diffusion is anytime we need to have something like a protein to open a channel. Or sometimes we'll use the term gate. So you'll see both of those. Opens a channel or a gate. Active transport oftentimes uses what we call pumps. So these are similar to channels, except they actually have a spot where ATP is kind of attached or utilized and will pump, and they always do this against the concentration gradient. So let's look at these in a little bit more detail here. So passive diffusion, just like with osmosis, is going to work in situations where we're moving from an area of high concentration to an area of low concentration, meaning when things need to spread out, we can use this simple diffusion method, and again, it has to be very small. And these things are going to spread out until they reach equilibrium. So let's say I've got a bunch of cows on one side of a fence here, okay, and I don't have any cows on the other side of the fence. Say maybe I have, let's just say, we'll call it 6 cows. I should draw 6 cows here. Oh, that'll take too long. Okay, no, 6 cows. With simple diffusion, what would happen is I would end up with, 3 cows on this side of the fence and 3 cows on the other side of the fence. And that would be equilibrium. That just means we have equal amounts on either side of whatever membrane we're talking about. So where in the body does this happen? Well, the big one that we use as an example is in the lungs. So you've got, I'm going to draw your little alveoli here. It's the little air sacs in your lungs, right? And you've got oxygen that comes in. And then around those alveoli we have capillaries. Okay, we have blood vessels, right, that wrap around them. So this is my bloodstream. And I don't have very much oxygen left in here, right? This is deoxygenated blood that's already distributed throughout my body. Well, what's going to happen is simple diffusion, and this does not require any kind of energy. So whether I'm making ATP or not, doesn't matter, right? We can do CPR and ventilate our patients, and oxygen will still get into the blood, even if their other cells aren't doing so high, right? So how much oxygen is going to come in? Well, it's going to balance it out. So let's say I have 2 oxygen molecules here. Okay, I can send one over here, and now I've got 3 in the blood and 3 out here. So I've reached equilibrium, right? So what does this mean, or why is this important for us? Well, think about the fact that the amount of oxygen that the alveoli contain is directly proportional, or that we could say the percentage of oxygen that the alveoli contain is directly proportional to how much will end up in the bloodstream. So normal room air is about 20% oxygen. Okay, so maybe we can say, I don't know, maybe that's the situation I drew here, where there's 3 oxygen molecules that'll stay in the lungs and 3 that end up in the bloodstream. Well, if I just put my patient on oxygen therapy, let's say like an oxygen kennel, normally we start that setting at about 60% oxygen. Well, what does that mean? It means more O2 in the bloodstream. Okay, than if they were just breathing room air. Why does that happen? Why does the blood suddenly take in more oxygen? It's because there's more oxygen on the other side of the fence. There's more cows on the other side of the fence. And so we can actually use this principle to force the body to take in more oxygen, which, let's say I'm in a situation like I have I don't know, pneumonia, or I've recently been in shock and my tissues haven't been perfused very well. I haven't gotten oxygen sufficiently to those cells. Well, we can kind of say, you know what, let's take in some extra. It'd be great if the body was smart enough to just take extra from the room air, but it's not, because it's simple diffusion. It's going to reach equilibrium. So the more oxygen we offer, the higher concentration, okay, the more will end up in the bloodstream. We use the same principle for anesthesia when we pre-oxygenate patients. We let them breathe in pure oxygen for a few minutes, and it allows them to store extra oxygen in their blood. So if while we're intubating or inducing, they don't breathe for a few minutes, that's okay. They've got some extra oxygen in their blood because we hacked this idea. We touched on this a little bit already. Facilitated diffusion is when we require a protein, or you can think about it as a key, okay, to open the lock, and the lock being the channel, to allow a specific substance into the cell. So this is the most important example in the body, and this is the one you'll see everybody reference. This is a mechanism that some electrolytes move by as well. But very, very important, since glucose and oxygen are the 2 things that the ATP require. Oxygen, we just looked at, gets in by simple diffusion. Glucose requires the help of insulin, requires the help of the key to get inside the cell, or else it just sits out there. Think about what a patient looks like if they're diabetic. If they don't have enough insulin, they're hyperglycemic, right? They have high blood sugar, and that's because that sugar sits outside the cells in the blood instead of inside the cells. So even though it seems like they have plenty of glucose available for energy, they actually, inside the cells, they would be hypoglycemic, right? They don't have enough inside to be able to do what they need. So in a normal patient, insulin goes and it binds to that insulin receptor. We said that opens the channel for glucose, and glucose is able to make it inside the cell. And a normal healthy pancreas is going to make just the right amount of insulin for the amount of food or energy the patient's taking in, so that enough of the glucose stays in the bloodstream to be available if the brain tissue suddenly need more, or you all of a sudden kind of run a marathon. Your body has other mechanisms we'll look at too. For instance, your liver stores glucose so that if needed, all of a sudden it can release it, but insulin is absolutely required here. Now, we've talked about, or will talk about in other classes, how sometimes we can also hack this mechanism, which is really cool. So you may be familiar with kitty cats who get a urinary obstruction. They may have urinary crystals or just mucus. You have that homework assignment and lab procedures that pertains to this. And what happens with urinary obstruction is the patient can't urinate, right? So no pee comes out, and as a result of no urine coming out, we don't get rid of the waste products that the kidneys would normally excrete either. The big one being potassium. Okay, the kidneys have to excrete potassium, and if we retain too much of it, it's very, very dangerous. So sometimes if we have a pet that can't urinate, and we're maybe working on placing a catheter and they have way too much potassium hanging out in their bloodstream because their kidneys haven't been able to excrete it, one of the things we can do is we can say, you know what, potassium, would you like to kind of be shoved away for a little bit somewhere where you can't do things like hurt my heart? Let's give the patient some insulin. And when we do that, it opens this glucose channel, and potassium will actually take advantage of that and move inside the cell. Eventually it'll come back out, but by then we'll have dealt with the urinary obstruction, and the kidneys will be working again, and everything will be fine. So sometimes we borrow this idea to be able to put things inside the cells temporarily. And I think that's really, really cool. Lastly, we've got our active transport, and we did say this requires energy, so we do have to have ATP available for us. Things have to be going pretty well in our bodies, and this is going to happen against the concentration gradient. So let's say we have 6 cows on this side of the fence and 2 cows on this side of the fence. Okay, with active transport against the concentration gradient, we're going to move all the cows to one side of the fence. Okay, even though it seems like, they should spread out. No, that's diffusion. That's passive transport. With active transport against the concentration gradient, we're putting them all on one side of the cell membrane or into one tissue compartment. The most important example for us is the sodium-potassium pump. So sodium is primarily meant to stay outside the cell. It's what we call an extracellular substance. It should spend most of its time, or most of the sodium in your body should be outside the cell. There's some inside, but most should be outside. Potassium primarily should be intracellular. It should be inside the cell. If it's outside the cell, we can see negative effects on muscle contraction. We'll talk about those more as we go along here. But for now, we need to know sodium outside, potassium inside. So what does that look like? Well, I've got a bunch of sodium, let's say, inside my cell. And I've got some outside my cell too. And same with potassium. Draw 3 little guys out here. Okay. And the sodium-potassium pump is actually going to make sure that these potassium ions move inside. Okay. And these sodium ions move outside. And because it is against the concentration gradient, this mechanism requires ATP, and they kind of trade. So a certain number of potassium will exit, a certain number of potassium will enter, and a certain number of sodium will exit. So this pump works all the time to make sure that these guys balance out. And that's particularly important because they do find other ways in and out of the cell. So they do still sometimes want to passively diffuse, or we saw in that last example with facilitate diffusion, they sometimes use other channels to get inside the cell. So this pump has to work all the time. It's kind of like having a leaky boat with our semi-permeable membrane. This pump has to work all the time to maintain this balance. Well, what does that mean for us? Let's think about it for a second. We said this requires ATP. Well, what do we need to make ATP? We've got to have enough oxygen. We've got to have enough glucose. So let's say I have a patient that is in the state of shock. Okay, maybe they have an anaphylaxis and their tissues don't receive enough oxygen for a period of time. Well, maybe then I don't have enough ATP, and maybe then, okay, I can get these levels of sodium and potassium out of whack, and that can definitely be life-threatening. So we're going to look at electrolyte issues shortly here, but all this stuff is connected. That's what I think is really cool about it. So we're going to start to think about how some of these principles that we've talked about of fluid movement and electrolyte movement affect our body on a larger level. We've kind of been looking at individual cells for now. The first is in terms of what we call fluid compartments. And the two big fluid compartments, or places in your body that can hold fluid, are the extracellular and intracellular fluid compartment. Sometimes you'll just see this ECF and ICF terminology used. They love to put it in articles to be confusing. Intracellular fluid is going to be 40% of our body weight. It's most of the fluid in our body, and that's just what's in the cytoplasm. Boy, this is an annoying background to write on here. So the cytoplasm contains about 40% of our body weight in fluid. It's kind of funny, we don't think about it, but when we're dehydrated, it's actually our cells that are kind of shrinking that don't contain enough fluid. It can be our other tissues as well, but serious dehydration is going to affect that cytoplasm. The other 20% of our body weight is going to be in what we call extracellular fluid compartments, and that's going to include interstitial fluid, which is the fluid between our tissue layers. We test this, right? We try tenting the skin and the shoulder blades, and if it stands up, it means we're dehydrated. That's because there's a lack of interstitial fluid to lubricate between those layers. Intravascular fluid, this would be blood. okay, what we think of as blood plasma, right? And then we have some other compartments here, our lymphatic fluid, our central or cerebral spinal fluid, so the fluid that surrounds our brain and spinal cord, and then also our joint fluid. You've also got fluid in places like your thoracic and abdominal cavities in small quantities, just to lubricate so your organs don't stick to the side of the walls. That would be not good. Now, how does fluid move between these compartments? Well, we already learned that. It's simple diffusion. So let's say I do something like give IV fluids to a dehydrated patient. Your standard lactated ringer crystalloid solution spends usually less than 20 minutes in that intravascular space before it's going to move through simple diffusion. And remember, with simple diffusion, we move with that concentration gradient. What does that mean in this case? Well, wherever fluid is missing, okay, it's going to spread out to those areas. So that's going to be something that we want to have happen, right? We want to go replace whatever the fluid in the cells that's missing is. We want it to move from the intravascular space to the intercellular space, or to the interstitial space. So simple diffusion with the concentration gradient. Now sometimes we're in situations where we don't want the fluid to move out of the intravascular space. Let's say I have low blood pressure. If I give fluids and it moves right out of that blood, then it's not going to necessarily help me with blood pressure. So that's when we start to give more specialized fluids, like our colloid fluids. But that's not what we're concerned about today. Those still move by simple diffusion, but they have ways of being a little bit more slow. So what drives the movement of fluid between the different compartments? That's the principle we talked about before of tonicity or solute concentration, and mainly we said this is sodium that's going to be the big driver here. So we're going to see that there's a certain amount of salt inside the cells. Okay, so let's say I'm a happy little cell here, certain amount of salt, and maybe I'm dehydrated, so I lost one of my water molecules. And I go ahead and I have a solution that I give the patient. Okay, that's salt and water out here to try to rehydrate them. All of our crystalloid solutions do contain solutes. That's why they're called crystalloids, is because they contain things like salt that are crystals and are dissolved in here. Okay. And the body's going to look at this and say, okay, well, we're moving simple diffusion with the concentration gradient. So out here, every sodium has a water molecule. We've got a couple extra water molecules. So let's move some of this water in here so the balance is the same, right? So that's how tonicity or saltiness drives the movement of fluid through these fluid compartments. And maintaining that water balance, we sometimes call maintaining osmotic pressure. So osmotic pressure is water balance throughout all of these different fluid compartments. There's another term sometimes you'll hear called oncotic pressure. And that actually refers to protein balance between these different fluid compartments. So that's the other driver here. Tonicity mainly, saltiness mainly controls water balance, but we do sometimes see that proteins, and we'll get to that in other lectures, but proteins can also affect this. So when we say something is isotonic, we mean it is the normal saltiness. Iso means normal, or the same saltiness as what's in the cytoplasm of the cell. And if we give a solution to patients that is isotonic, it's going to be the same saltiness as the cells. It encourages rehydration. It encourages simple diffusion, evenly distributed across all the fluid compartments. If we give a solution that's hypotonic, so less salty than our body cells and tissues, just, distilled water would be an example. It's going to encourage water to go in and dilute that sodium that's in the cells, so everything balances, right? So that the lack of saltiness outside balances with the lack of saltiness inside, and that'll cause the cells to swell and burst. So if we, let's say, did give distilled water to our patient's IV, we would get massive hemolysis and death. Not just the blood cells would rupture, lots of our body cells would rupture, but the hemolysis would probably kill us first. If we give a solution that is more salty than the cytoplasm, based on the principles we're talking about here, okay, what's that going to do? Well, there's a bunch of sodium out here, right, because we've given that hypotonic hypertonic solution and not a lot of water out here, then water will be encouraged to move out of the cells. So it's going to shrink the cells. And we do occasionally do this to try to do things like reduce edema in the tissues. We give it in the case of head trauma to reduce brain swelling. They give it for cows for mastitis. We can also give it, let's say I have a patient that is in shock and needs more fluid in their blood vessels. We want to take fluid out of the cytoplasm and borrow it, let's say, into the intravascular space. We can do that by giving a hypertonic solution, a saltier solution than what's in the body cells and tissues. We will have to go back and replace it eventually, though, with one of our isotonic crystalloids. So this is kind of a cool thing that we can play on as well. So when we look at these different fluid compartments and ask ourselves, how does that fluid move from the intravascular space to the intracellular space, okay, or how does it move from the interstitial space into the cells or out? This is all going to be based on tonicity, so salt levels. Now, homeostasis is a term we kind of bandy about. Homeostasis is maintaining all the conditions in a stable way so that we can perform the chemical reactions that we need to do things like create ATP, which we use for all of the other processes in our body. So we need consistent conditions. So I kind of think about this as like running a snap test, right? It can't be too cold, it can't be too hot. In the room, we have to have certain conditions or the test doesn't run properly because the snap test is a chemical reaction. We try to get that color change or that dot to show up on our snap test. So it's the same here for homeostasis. Homeostasis is going to be a condition or a set of conditions we need. So temperature is a big factor here. We've got to have the right temperature in our body. We need the right pH in our tissues and our fluids. We need to make sure we have enough, we said, oxygen, and we have enough glucose available. And there's probably other factors that you can add here too, but these are the ones that we're going to really focus on, especially that pH level for the rest of our conversation here. Before we look at blood pH regulation, we do need to take a little detour into electrolytes. And electrolytes are ions. An ion is something that's on the periodic table, right? It's one element. It's not something like a protein that's a complicated. It's one particular little molecule, and it has a positive or a negative charge. And we call those positively charged ions cations, negatively charged anions. And I always think of anions as like onions as negative because you're crying. That's kind of how I remember here. The ones that we really want to know are listed here. So we've got sodium, Na, and potassium. Those are both cations that are important for us. We've got chloride. It's our most important anion. And then those are the ones that are going to be circulating in our blood and in our cells that we spend most of the time measuring on our lab tests and talking about the effect in their body. We do have some others that can be significant that are primarily found in bone tissue. We do have some in our blood as well, but they are stored and should spend most of their time in bone. Calcium is a cation. We've got phosphorus is an anion since it has a negative charge, and then magnesium. So let's look at how each of these are important for us. So we already said that sodium should primarily be in the extracellular space, and potassium should primarily be in the intracellular space. And it's good to remember that sodium and chloride hang out together, right? That's what table salt is NaCl. And it's the same in the body. They kind of like to hang out, so they're friends, so they're both in the extracellular space. Now, sodium's main job we already touched on, and that's to regulate water balance, okay, or regulate osmotic pressure. That's all that means is water balance, or where the water is in your body. And this is absolutely critical to maintaining our blood pressure. So if we don't have adequate amounts of sodium, or we have too much sodium, we're going to have derangements in our blood pressure. You might have heard, if you have high blood pressure, you want to avoid salt. Well, this is why. It's going to encourage the water to move out and increase your blood pressure if you have more sodium in your blood vessels, let's say. So sodium has this very important function of water balance. Now, you might have heard there was a case years ago where someone got water toxicity, right? She drank gallons and gallons of water because she wanted to win a Nintendo Wii, I think it was. This was like 2006, 2007. And she died. Well, how do you die with water or too much water in your body? Well, she didn't die from too much water. She died from too little sodium. So too little sodium would be hyponatremia. Okay, and how does that kill you? Well, too little salt is going to disrupt your water balance, and you'll actually get pretty significant swelling of your tissues, and you'll get particularly swelling of the tissues of your brain. So that's usually how you die of hyponatremia, okay, is what you're going to see there. Now with chloride, chloride's main job is to form hydrochloric acid, or HCl, in your digestive tract. Okay, hydrochloric acid is going to be formed in your stomach. It's used to digest your food, and then we actually reabsorb it. So the chloride goes back to your blood. So this is recycled over and over and over again. Derangements of chloride are not usually life-threatening. We'll look at those here shortly, but if you do have, let's say, a situation where you're vomiting a lot, you might get hypochloremia. And why is that? It's because that stomach acid is not being reabsorbed into your bloodstream, and that chloride's not being recycled, and instead it's being, vomited out, so we don't have it anymore. Now, potassium is probably the biggest or most important one that we see sometimes as an issue for our patients. Potassium is required for proper muscle contraction. And if these levels get too high or too low, so we get hypo or hyperkalemia, so remember K, like potassium is K plus, right? Kalemia, then we'll see that we have trouble with muscle contraction. And remember, the heart is a muscle. So patients with hypo or hyperkalemia will get life-threatening cardiac arrhythmias, and that'll be their cause of death. So we might have to do something like borrow insulin. Remember, we said borrow that mechanism to deal with the situation. As I said, the other electrolytes are usually less important or less likely to cause us a problem, but we do see issues from time to time. Calcium, we have to have adequate amounts of calcium available in our bone and then available in our blood to be able to properly relax our muscles. And it also has a cardioprotective effect. It helps our heart be protected from the influence of other electrolytes. So we do see situations where we don't have enough calcium, and I'll include a video in the playlist here, but eclampsia. It's a situation where you might have a dog or a cow. For my practice, it's usually, small dogs that maybe they're Chihuahuas and they have 3 enormous puppies, and then they lactate and they nurse those puppies. And when they do so, there's calcium in the milk, and maybe they haven't had a lot of calcium in their diet, so they don't have a lot stored. Well, what we see then is eclampsia or hypocalcemia, where they no longer have enough calcium available to them. And the big thing we'll see is muscle tetany and muscle stiffness. And that means they're not going to be able to contract their muscles, and eventually, same thing as we saw with potassium, if we have a problem with our muscles, we get cardiac arrhythmias. So this can definitely be life-threatening if we don't have enough available to us. Phosphorus is more involved with building bone and teeth and tissue repair, so very involved in healing. Usually we don't see derangements in phosphorus unless we have crazy stuff going on with our other electrolytes. And same with magnesium. Magnesium is required for ATP production, but unless we have a crazy electrolyte disorder or some of our endocrine disorders, these are not usually going to be issues for us. They are things that we can test for. And if we have patients that have other complicated conditions, sometimes we do, because they can be a contributing factor. So for instance, complicated diabetic patients sometimes get derangements of phosphorus and magnesium. But in general, these are not going to be like, oh no, my patient's dying of a phosphorus deficiency. That's not typically what occurs. Lastly, we're going to look at pH regulation. So within our bodies, within our blood and our tissues, we have a very narrow range of pH that we need to maintain. You definitely do want to know this for the test, 7.35 to 7.45. And we don't have a lot of margin for error here. The scale for pH is not what it appears to be. So when we go from 7.35 to 7.45, it's an exponential change. It's not just, 0.1 higher. It's a big significant difference. So this is actually a fairly wide range, and we can't tolerate pH outside of here. This is different than urine, right? We said urine could have a big range of 6 to 8. Well, I'll tell you, I see when we run blood work on patients that we're doing, let's say, CPR on, their pH might be, 7.1 kind of a thing, and they're dead. I've seen patients alive with like 6.9, but that's barely survivable. So this is not something that we can play around with. Unfortunately, our body has a really sophisticated system to be able to always make sure that we're in here. So I mentioned when we talked about urine that your body changes the pH of the urine all the time, partly to get rid of things that are too acidic or too alkaline, to be able to balance this so that the blood can be in this very narrow, very precise range. So sometimes you'll see people online say, like, drink a ton of orange juice, it'll make your body more acidic. Well, I certainly hope not, because if it makes your body more acidic, you're going to die. So that's not a great plan. And also, unless your coping mechanisms, your internal compensatory mechanisms are not working properly, you won't be able to drink enough orange juice to affect this. So hopefully that'll be the case anyway. All right, so when our pH decreases below 7.35, not good, because those chemical reactions, like the ones that produce ATP, aren't going to be able to happen properly. We refer to this as acidic, or you'll see acidosis. Sorry guys about the red pen, I can't figure out how to make it a different color for these recordings, but the other backgrounds aren't this bad. So acidosis, and that means we've got either a high concentration of hydrogen ions, or we've got a buildup of a lot of anions. So most of the processes in the body, so ATP production, cellular respiration, all that stuff, produce acid. So acidosis is going to be a much more common problem than alkalosis, which is what we see if we get too alkaline. So the big one is going to be CO2. Okay, CO2 is an acid in our body, and of course we get rid of it through respiration. And if we make it as a result of cellular respiration, then we get rid of it through our actual respiration in our lungs, right? They call that external respiration. And if we don't get rid of it, if for some reason we're not able to exhale enough, our CO2 builds up, and CO2 acts as an acid, so we would get an acidosis. Now, if we have too many cations built up in our blood, okay, or a low concentration of hydrogen ions, then we actually go the other way. We get alkalosis. So we'll look at some of the reasons why this might happen, our acidosis or our alkalosis here. So one of the most common reasons that we see acidosis is that the kidneys don't remove enough of the acids, enough of the hydrogen ions in our blood when they're doing their thing. So renal disease is a situation where our nephrons are not making good decisions about what to lose and what to keep. And if our kidneys are not removing enough of the acids, the waste products in our blood, out into our urine, then we get metabolic acidosis. We get acidosis as a result of a problem with our metabolism. Now, another situation we can see is that buildup of lactic acid. So anytime my cells and tissues go without oxygen, we use the example of running up and down the stairs where I don't get oxygen to my muscles quickly enough, but let's say I'm in shock because I have parvo, and maybe I'm in hypovolemic shock. I'm really dehydrated, so I don't have enough blood volume to move oxygen around my body. Those cells switch over to anaerobic metabolism. And when they do that, they build up, or they produce lactate and lactic acid as a result. So any state of hypoperfusion, or lack of oxygen, right, to the tissues, lack of O2, is going to result in the production of acid in the form of lactate, and that will eventually, if there's enough of it, cause metabolic acidosis. We see another situation with our diabetic patients. I'm going to draw up here because the color is driving me crazy here, where if they can't get enough glucose in their cells, because remember with diabetes we're not making enough insulin, okay, so then the cell's like, well, what do I do for energy? Okay, how do I make ATP, because that's what they're trying to do, if I can't use glucose? Can't get into the cell without insulin. So the cells start to use fat for energy, okay? And when they use fat for energy, the process produces ketones. And ketones are an acid. Now, your body can do this in small amounts. You've probably all heard of a keto diet. It can take fat in small amounts and use that for energy instead. But in these diabetic patients, it has to use fat exclusively for energy. Even if you're on a keto diet, your body's still making insulin and still using some glucose. These diabetic ketoacidosis patients, the keto comes from making ketone bodies, or these acids. Acidosis comes as a result of what we see here. They exclusively have to use this fat, and then they just make tons and tons and tons of these ketones in the process of making ATP. It makes their blood super acidic. So that's our diabetic ketoacidosis. That's going to be a problem. And we'll need to go ahead and help them to start using glucose for energy again to resolve this. Now, respiratory acidosis are situations where we can't excrete enough CO2. So think about, maybe I have pneumonia, and in my little alveoli, there's a bunch of mucus in my lungs, and maybe the CO2 then tries to get out but isn't doing a very good job of escaping. Well, that CO2 is going to build up in my blood, okay? And then I get a respiratory acidosis. Alkalosis, we said, is less common than acidosis. We see two main reasons why we might get alkalosis. So the first is vomiting. If we lose a bunch of that chloride, and remember chloride is an anion, so it is kind of an acid normally, and we should keep it. Our hydrochloric acid is recycled back into our blood. Well, for vomiting, we lose a ton of chloride, then we'll see that we get a metabolic alkalosis. And if I have that patient come in, let's say that has a foreign body, and he's been vomiting all day, and I check his blood pH, okay, maybe it's going to be, instead of 7.45, I'll get 7.5, because his body's lost so much chloride, so much stomach acid in that process of vomiting. Sometimes the same can be true for dehydration, especially if it's accompanied by vomiting. Now, respiratory alkalosis would be hyperventilation. So let's say I've just been hit by a car and I'm breathing super fast to get rid of, I'm breathing super fast not because I need to get rid of acid, but because I am in pain, okay, just as a symptom of pain. In that situation, I lose a bunch of CO2, and remember we said CO2 is an acid, more than I need to maintain my blood pH, and I might at least temporarily have a respiratory alkalosis. So in that situation, it's pretty easy, right? We just deal with the pain, we deal with whatever's causing the hyperventilation, and things will balance themselves out. Now, what are the ways that my body normally compensates or keeps my pH in the normal range? Well, we call compensation in this case buffering. Buffering is maintaining that blood pH within that 7.35 to 7.45 range. And your body does this all the time. It's why you can drink that whole gallon of orange juice and not change your blood pH. And the first way that your body buffers is by producing something called bicarbonate. And bicarbonate is a substance, it's a chemical, it's something that we sometimes have on the shelf that we can give as an injection, and it's something that your kidneys make. So your kidneys make this substance called bicarbonate. Bicarbonate acts as both a weak acid and a weak base. And what's cool about that is that it can either make your blood more acidic or more alkaline, whichever your body needs. So it can perform either function. So your body produces this chemical all the time, and you will not see a derangement in your pH, so we won't get out of this range unless, let's say, your kidneys aren't working properly and they haven't produced enough bicarbonate, or we've run out because our acidosis, maybe if we're hypoperfusing, we've produced so much lactate that the amount of bicarbonate we have available to cancel it out has run out. So that's what's kind of cool about this bicarbonate. Up until that point, it can do either job if it needs to. Now, I mentioned this is a drug that we can give, and we do have on the shelf, so sometimes if we notice, especially a metabolic acidosis, we can give some of this bicarbonate and help the body out. The other way that we buffer our pH is through respiration, and we've already said that CO2 is an acid. So if my blood pH, let's say I've got acidosis, okay, I can breathe more. So I'll get rid of CO2. Okay, if I have alkalosis, my body can say, you know what, breathe more slowly. Hold on to more acid. We have an alkalosis, we need to keep more acid to balance it out. And so all of this happens all the time without your thinking about it. I like to think in A&P about how many different ways I would die if I had to consciously control all these processes, and blood pH is a pretty good example, right? I can't think about how often I need to breathe to maintain my blood pH. That would just be ridiculous. But your body uses both of these buffering mechanisms all the time, without your knowing about it, to be able to maintain this within the normal range.